It's been a while since I've done this. It's biology with Mr. B. Biology with Mr. B. That's me. So, hello. Hopefully, I won't have to do too many lessons like this, but this is the position we're in. So, let's rock and roll. This is actually the first lesson of Module 6. How exciting! And in Module 6, the focus, or at least the main focus for my half of the lessons, is everything genetics. Starting with how you can control whether your genes are switched on or switched off. Because we are eukaryotes, we are multicellular. Every single cell in our body is derived from that original zygote, which means every single nuclei in our body is identical, has exactly the same chromosomes, exactly the same genes in the same loci, everything. The only thing that makes certain genes, well, certain cells differentiate, makes certain cells do some jobs, other cells do different jobs, is because sometimes genes can be switched on or off permanently. That's differentiation. Other times, and if you just think like an individual cell, like a salivary gland cell, you don't want to be secreting salivary amylase constantly. No one likes a drooly sort of mess. We don't want to be drooling and just salivating constantly. You only want to secrete the salivary amylase, and therefore you only want to make the salivary amylase when either neuronally or hormonally you're told to. An element of sort of communication, neurons or hormones. Obviously, salivary amylase, that would usually be neuronal. You look at the food, you go, rah, 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 and you start salivating. One of those. So, in this topic of cellular control, it's effectively me trying to explain in a sort of real-time, real-world situation how we can affect the genes we already have in our cells. How can we switch on, switch off, promote, inhibit genes that our cells naturally have, and why that might be useful. So I always enjoy these four. It's a very small topic, small booklet. If you've not got the booklet and you happen to be passing by biology prep room, please do knock on the door and just say, "Hi, please, can I have a cellular control booklet?" That was a bad impression for you guys, but that'll do. You've got 2 spec points that are nice to learn, fun. Types of mutations, which in all fairness, you probably already know what mutation is, and you probably already appreciate the effect it would have on like the triplet code and protein production if you mutated a base. Down here at the bottom, this is how you destroy a cell. How do you destroy a cell on purpose? How can you, in a controlled way, destroy a cell? And it's a process called apoptosis, and why that is really, really important, and why mitosis is also important in development of our body form. The bit to panic about is the bit in the middle. I say panic. I promise I am going to teach this in a way where you will find it that it's okay. But I may as well warn you. I may as well go into this as if it's the hardest thing you're ever going to do, because when it's not, you'll feel good about yourself. So B can be brutal. The regulatory mechanisms that control gene expression at transcriptional, post-transcriptional, post-translational level. This one and this one, transcriptional and post-translational, are actually relatively brief. There's not much going on there, but there's like a key idea. Post-transcriptional, we do have to look at an example called the lac operon. The lac operon is obviously completely new to all of you, and therefore people find it challenging. I promise I will teach it in a way that you won't find challenging. But please, when we enter our lac operon lesson, our lessons after mutations, please be prepared that it might be really challenging and you might not grasp it straight away. Spec point C is one that even if you don't actually grasp what's going on, I'm actually not worried. You might remember Mr. Barber is the chief examiner at OCR, and his feedback from the examiners is that not to worry too much about this spec point because the examiners don't understand it either. don't get me wrong. Of course, I'm going to teach it in a way that will get you what you need to get out of this. I'll get you the marks. I'll understand it in the simplest way. I'll make sure everyone has that. And I will try my best for those who are destined for A stars to actually justify why things are happening the way they are. And I'll give you the full beans. But for the vast majority of students, just knowing the simple key facts is all you're ever going to need to access marks in the exams, because they don't seem to do any questions on this particular spec point because it is so challenging. And yet the examiners aren't too sure either. Anyway, let's go. So we are today looking at gene mutations and their possible effects on protein production and function. So we got some keywords: substitution, insertion, and deletion. I'm actually going to add one in, but I'm going to add it in relation just to these two words. It's called Indel. You can probably see where I get the term Indel from. In is from insertion, del is from deletion. Indel. So substitution and Indel. I'm actually going to add another keyword to this one. I'm going to call this point. Point. I can hear the Alexa going in my house. That'll be quite funny if you suddenly get an Alexa of my daughter, sort of like announcing to the world she's hungry. I don't know. So, this recap and DNA protein synthesis, I'm going to do a small bit, not as much as I would have done in school, but it's probably better I do a small bit whilst I'm at home. And then I'm going to describe what these keywords are, what point, what indel means in the terms of mutations, and how that would affect proteins. So let's start. This is a Foundations of Biology Year 12 recap. Firstly, every amino acid is coded for by a triplet code of bases on your messenger RNA, called a codon. So if this is at the ribosome, if this is our messenger RNA, so let's say messenger RNA would have U's, wouldn't it, instead of T's? Let's go ACU. And this one would be, which one haven't I used yet? GG. Those greens were A's, the yellows were C's, the reds were U's. There you go. So that's on messenger RNA, and there's a whole entire sequence called translation, where you've got a transfer RNA molecule coming in, and with its complementary anticodon, it binds to that first triplet code. And on the other end of the transfer RNA molecule, you have a specific amino acid. And we can actually see this with our different shaped amino acids, because here I have a triplet code of ACU, and I've made a circle amino acid. So a circle amino acid being brought by my transfer RNA. Down here, I've got another ACU, and again, I've made a circle. GGA is a different triplet code, a different codon. I've made a triangle. And CUA, again, is a different codon, and I've made a square. There are three features of your genetic code that are essential to appreciate as we move into mutations. The first of which is that our code is degenerate. What that means is that there are usually more than one codon which codes for every amino acid, and I can explain that. If you think of your triplet code having 4 bases, that's actually 64 possible codon combinations. And yet we only have 20 amino acids in humans. So therefore it would make sense that for every amino acid we produce, there's potentially at least 3 different codons that would make the same amino acid. I can't remember if this is entirely correct, and I might have deleted the part of this PowerPoint that had the actual answers on, but I have a sneaky feeling it's something like, if it's ACC and ACT, this is the DNA bases, they would both code for the same amino acid. I think it's asparagine or something like that. So that's what we mean by degenerate. Different codon, yet same amino acid. Second idea is that this code is non-overlapping. You only read your triplet once. So that triplet gets read and then it's done. Then that triplet, then that triplet, and then that triplet. You're not mixing, you're not maxing. It is very uniform. 3, 3, 3, 3 bases as you go. And it only gets read once as you make your amino acid sequence. The final is the universal code or near universal code. If ACC in a human codes for asparagine, ACC in a plant will code for asparagine, in a bacteria will code for asparagine, in a fungi will code for asparagine, even, I don't know, even in your, I don't know, any sort of organism in your wildest imagination, ACC would be asparagine. That's the near universal code. I said it, I thought I might as well eat it. No, it is right there. Of course it is. Sorry for that bing. That's Mrs. Bateson being loud, as you'd expect. So, exam questions. So let's imagine our DNA strand is that ACG, TTG, GTG, CAC, GTG. Good for it. Notice how this codon here has the T's, so we must be referring to the DNA strand, not the messenger RNA right now. So if you had that strand, how many amino acids would it code for? Pause me. Whilst you do the question, make sure you've paused me. Play me when you've done it. So hopefully you've now done this. How many amino acids were there? There were 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So that would produce 2, 3, 4, 5 amino acids. However, it does tell us in the question, obviously, that is an amino acid, that is valine. But please bear in mind, sometimes you can have stop codons at the end, which don't code for anything. So always be careful if you're dealing with like a DNA strand. If it's the entire gene, then the last codon is actually a stop codon and doesn't code for amino acid. So just bear that in mind. On this one, we know it does because we've got the table. So what sequence of amino acid will the section of DNA add to a protein? So ACG was threonine, TTG was leucine, GTG, GTG was valine, CAC was histidine, and then it was another valine at the end. Okay, so that's our 5 amino acids that would add to that protein. Mutation time. A mutation is a random change to the genetic material. So that could be a change in one of the bases, or it could be an entire change to a chromosome. Today's lesson, I'm only focusing on a random change to genetic material. I only want to focus on if you change a base, not if you change an entire chromosome. So that there is our definition. Okay, so let me just get rid of this. I'll do all this blank bit here. Imagine we have a sequence of DNA, which is cat, 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 cat. Because who doesn't like cats? So I've clearly got 1, 2, 3, 4 codons, and I'm going to make 4 amino acids. And because it's the same triplet code, I'm going to make the same amino acid, aren't I? And let's say, I don't know for sure what cat is. Let's just say it's glycine just for fun. So obviously this would be transcribed into messenger RNA. That would be then read by the transfer RNA molecule, an anticodon at the ribosome. And let's just say we do get glycine, glycine, glycine. There are a few different ways we can mutate. A few different ways that we could randomly change this base sequence. This is G for glycine. The first way is What if we just have a swap? What if during DNA replication something daft happens and instead of cat, it reads cat, cack, cat, cat? I mean, that's possible. The difference is obviously this one here. It was a T, now it's a C. So it's a simple, what we'd call a simple substitution. I'm just going to write it up here as an example. We have sort of substituted a C instead of a T. Completely random chance. We know from the principle of semi-conservative replication, this should not happen, but every now and again something daft does happen. Think how many cells are dividing your body every day, how many millions. There's bound to be a few mistakes. No, nothing's perfect. So in terms of what this would do to the amino acid structure, well, that would still be glycine. That would still be glycine. That would still be glycine. And this one, again, not knowing off by heart, although I think we did look at CAC last time, not knowing off by heart, I don't know what this is. This could be a different amino acid. Let's say this one could be, I know it wasn't valine, but let's just say valine. If you, by the way, did just remember which one it is, well done, because I can't. Let's say it did code for a different amino acid. And then these amino acids try to make their folds, make their bonds into the globular protein. It may be that the introduction of this amino acid could be harmful. It could make a protein that doesn't function. You never know, it could be beneficial. It could make a protein that's even better at functioning. Maybe it improves the induced fit model of enzymes. Or it might do nothing. The protein might still fold in exactly the same way, and you get exactly the same result. Not only that, let's imagine for a second that because of the degenerate code, let's imagine CAC also codes for glycine, in which case it is a mutation, but it's done absolutely nothing to the amino acid sequence because our code is degenerate. And sometimes you can have a different triplet code, yet get exactly the same amino acid. So mutations aren't always that bad. So let's look at some types. So most commonly, if you're going to get a random mutation in DNA, it's going to be during replication, during the S phase of interphase. And even then, if we're thinking of like what's damaging, what's not, well, a mutation during mitosis isn't going to be as serious as a mutation during meiosis. If I have a mutation during S phase of one of my cells as an adult, and let's say it's a really harmful mutation, and that cell, it's like one of the proteins for respiration no longer is the right shape, so it doesn't work, and that cell can't respire, and it's only one cell. And I've got trillions of them. One cell not functioning is not going to be damaging for me as a human being. Of course, there are always some horrible examples. Cancerous cells as a horrible, horrible example of a single cell being mutated that can do this and can cause harm in an adult. But mostly it won't. If you have a mutation associated with meiosis and making gametes, that's when you can have significant genetic disorder. Because if your mutation is in your egg cell, or it's in your sperm cell, as they fuse and make the zygote, the mutation is now in your zygote. And the only way that zygote can now develop is through mitosis. So every single cell after, every differentiated cell, every part of your development would contain that same mutation from that one random change in meiosis. So these meiosis-related mutations are the ones that are going to be the most harmful and or the most beneficial. This could be something really good that improves the individual and leads to natural selection evolution. So again, there's just something to bear in mind if you're discussing sort of harm benefits of mutations. If it's a mitosis division, it's likely not to impact the entire organism. It might impact the cell, and if you're unicellular, you're buggered. But if you're a human, it's just one cell out of trillions. If it's meiosis and it's your gamete, then that can be passed to the offspring. And if that goes to the offspring into the zygote, every single cell in that offspring will have that mutation. That could be incredibly harmful or incredibly beneficial. Or it might do nothing. It could be a completely neutral mutation that makes no difference. So, two types of mutations, two ways it can happen. The first is a point mutation. So, the one we did just before was a point mutation. We start with our normal DNA sequence of cat, 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 and the point mutation would be when it replicates, it's just a slightly different letter, a different base is substituted in for the old one. That's a point mutation. The second type is Indel, insertion or deletion mutations. What happens there is a little bit more interesting because instead of substituting a base, you actually either add or delete them. And it can cause something called a frame shift. Let me show you what I mean. So again, if we start with cat, 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 if I add a base in, let's just say I add an extra T, well, my first amino acid is going to be the same. My first codon's on the same, but now that's different. That's different. And I've got this random base here that's doing nothing. The reason why we also call it a frame shift is because where the mutation occurred, and the mutation here was in the fourth, it was this bit here, that was the insertion. Where this mutation has occurred, every single codon afterwards is now different. In a point mutation, because you're only potentially changing one amino acid in the entire gene, it might not cause any harm. The protein might still function exactly the same. An indel mutation, because every single codon after the insertion is changed, that means huge numbers of amino acids are different. It means it's virtually impossible for that protein to function correctly. And just to prove you get a frame shift with a deletion, if I just draw that in as well. So start with cat, cat, cat. If I delete, let's say I delete that T there, I end up with cat, so same. I've deleted that C, so we go straight to the A. It is now different. And I haven't even got a base there. So again, after the mutation, where the deletion was, and the deletion was here, that's where the C's missing. Every single codon afterwards is now different, which means we get different amino acids for all these subsequent codons. And that's a frame shift. Very harmful, potentially. Okay. The next thing, if you were in class, I'd then ask you to try and come up with what these different mutations mean. So hopefully, if you are just want that bit of like minute thinking time to try and predict, or if you're with a little group of people watching this together, have a little competition so you can get the closest to the actual definition. So pause me if you want to. I'll just go through. So if you... Okay, I didn't really look into it that way. Right, starting with nonsense. A nonsense mutation is just quite funny, really. Don't worry where it says find these on your table. Don't worry about that. A nonsense mutation is where it's a substitution. These are all, by the way, these are all point mutations. These are all substitutions. Where you've swapped out one of the bases in the stop codon. And it means the ribosome is never told to stop making its amino acid chain. Which means it just keeps making chain after chain after chain after chain after chain. And these aren't necessarily, it's not necessarily making the same protein again and again and again. It might be making different amino acid sequences of different genes and just putting them all together because nothing is telling it to stop. That's nonsense. Literally nonsense. You are not going to be producing a protein that functions because it's just going to merge with another protein that is coming after it in the ribosome. Silent. Missense. A missense mutation is where the brand new base results in a completely different amino acid sequence being coded for. So where it, in my example, I think I had cat and I said that was glycine, and if I changed the T to a C, it made a different one, valine. That would be missense. So you're actually making a different amino acid sequence, and therefore potentially a different protein in the way it folds into tertiary structure. Silent. This is the one that relies on your code being degenerate. A silent mutation is where, okay, you've got a different codon, but because of the degenerate nature of your code, you get the same amino acid regardless. So if your codons are GTC and GTT, that would actually be the same amino acid. So it's still a mutation. You've still got a random change to the DNA, but because it's exactly the same amino acid, it's therefore going to have no effect on the resulting protein. So yay, well done, degenerate code. It actually gives us a bit of protection against a mutation. So these are just some like little nice little works examples just to show you an example other than cat, 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 although this one does say cat at least once. So this is our point mutation, where you can clearly see one base has been substituted for another. Used to be A, and now it's G. And in this case, this is showing us a missense mutation, because I have got a different amino acid. An example could be sickle cell anemia. Sickle cell anemia is caused by a single point mutation in the gene that codes for the hemoglobin. And an A is swapped out for a T. It's a missense mutation because it is a different amino acid and you do get a different protein. Specifically, in sickle cell anemia, what happens to the hemoglobin is that when it's not bound to oxygen, it crystallizes. And that pulls the shape of the red blood cell into a bit of a crescent shape, a sickle shape. Apparently a sickle is something you use in like farming. I don't know. All because one amino acid was different, which meant different bonds were formed in the secondary structure for the alpha helices, which meant different folds could be formed to make tertiary structure with the hydrogen bonds, ionic bonds, disulfide bridges, hydrophobic, hydrophilic interactions that are essential for that tertiary structure. One amino acid has made that difference. Interestingly, it's not the only time we're going to look at sickle cell anemia in Module 6. When I start to talk about evolution, then I will link to it again, because sickle cell anemia is absolutely fascinating. If you are So sickle cell anemia, by the way, it is dealing with a recessive, well, it's recessive slash codominance, a bit of both, type of allele. Which means if you have a healthy dominant allele, you don't have sickle cell anemia, you don't have the crescent shape, so you don't have the dangers of that. But what you do have is a slight sickling, a slight change. And if you even have that slight change, it actually makes you more or less immune to malaria. So in places in the world where malaria is high, think Africa, the cases of sickle cell anemia have been increasing because it's actually an evolutionary advantage to have at least one copy to be heterozygous for this faulty allele. So it's effectively in a place of the world where malaria is high. If you don't have the fault, if you're not heterozygous, so if you're homozygous for the healthy alleles, you mean that you're more at risk of dying from malaria. If you're homozygous for the recessive faulty alleles, you're at risk of dying from sickle cell. But if you're that perfect balance in the middle, heterozygous, you can still transport oxygen correctly. You don't get the side effects of sickle cell anemia, but you do protect yourself against malaria. It's cool. And that's why sickle cell anemia tends to be more prevalent in people from Africa. So this is our Indel. This is one showing you an insertion. They've put a T in front of the cat. And that's given us a completely different, obviously, amino acid here. In the deletion, we've removed this A here. And again, we are dealing with completely different amino acids. These mutations are much worse because we get the frame shift. Every single triplet code after the mutation is now different. That's different, that's different, that's different, that's different. All further amino acids are going to be changed, including the stop codons at the end. So this mutation is massively disastrous for protein synthesis. So in terms of how we describe your mutations, if the protein still works and your phenotype is unchanged, we say it's neutral. So silent mutations are going to be neutral. Some missense mutations will be neutral if it doesn't affect the protein's function. If you're dealing with an indel mutation, it is likely going to cause a protein that isn't synthesized or doesn't function. And if that non-functioning is negatively impacting the individual, it would say it's harmful. Missense mutations, as we saw with sickle cell anemia, can also be harmful mutations. But this is how evolution works. If there's a change and it makes the protein have a new or a useful characteristic in the phenotype, it will be conserved. You'll be more successful, more able to survive, more able to reproduce and pass down this gene to your offspring. This is natural selection. So indel mutations, you could stumble across the great new protein that helps do something. Missense mutations as well could be beneficial. So never forget that. They could be beneficial. Without mutations, we would all still be single-celled organisms. Mutations are good, generally, or at least good for a species. Not always good for the individual, but good for the species to ensure genetic variation and ensure survival as our environment changes. Some examples of harmful mutations: cystic fibrosis. For 70% of CF sufferers, mutation is it's a deletion of an entire triplet. So it's a deletion of a codon. So effectively, you're just missing a single amino acid in that sequence, and that folds the protein in a way that makes the mucus that little bit thicker, stickier. The protein, by the way, is actually a channel protein. that exists in the walls of the goblet cells. Proto-oncogenes, which can be converted to oncogenes by a point mutation. An oncogene is a gene that promotes uncontrolled cell division. That's cancer. So a single point mutation can cause these proto-oncogenes to turn into oncogenes, which are genes that promote a cell just dividing rapidly, uncontrolled. And Huntington's disease is caused by a stutter, not the type of stutter I speak with, a repetition of a triplet, CAG. CAG, 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 CAG. We've all got these repeating sections of CAG, but if it goes above a critical value, and I think something like 15, the person will start to get symptoms. CAG, 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 CAG. So if you get 15 CAG, 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 CAGs in a row on this gene, then you develop the symptoms of Huntington's. Most commonly, and probably the best way we could relate mutations to our science that we've learned already is through enzyme activity. Because we've learned in so much detail how the shape of an enzyme has to be so specific in order for it to function, and we've learned about protein structure and therefore like the idea of tertiary structure being the wrong, essential for shape and the bonds within being essential, if we do have a mutation that changes our amino acids, we're going to get different folds and different bonds form in tertiary structure. We're going to get different shapes. And that's easy to talk about, therefore, like reducing enzyme substrate complexes and therefore slowing or even stopping enzyme activity. So this idea of linking it to enzymes is very much hopefully a go-to move, linking things you know about proteins, enzymes, and now mutations together. Finally, causes of mutations are by mutagens. Mutations are random. It is random chance whether you have a mutation. But a risk of developing mutation can be increased with exposure to mutagens. X-rays, gamma radiation, carcinogens in cigarette smoke, certain chemicals within caffeine, and UV lights are good examples, but not all, but are good examples of causes of mutations. And therefore, how do you lower your chance of getting mutations? You try to avoid those mutagens. Will it work? Not necessarily. People can go their entire life smoking 20 cigarettes a day and never develop the mutation that gives them cancer. There could be other people who actively avoid every single risk factor, and they might be the ones who unfortunately suffer first. Mutations suck because they're random and it's random chance. You can increase your risk, but that doesn't mean you'll definitely get those extra mutations. So, that is absolutely all I wanted to do in this lesson. Should I be off another lesson with you, I certainly am not allowed to return until at least Wednesday, it seems. And then obviously, if me or Mrs. Bateson develop symptoms, then obviously we would have to be off a bit longer, because obviously we'd then start our sort of a 7-day cycle, as if we get our symptoms and recover. Anyway, so whenever our next lesson is, I'm going to be setting you a few tasks in the booklet to do with mutations. It is as straightforward as that. It won't be a recording like this. But thank you very, very, very much for accessing this recording. You know, hopefully by now, I am literally available on Teams any time of the day. That's also, it's more likely to get me by Teams, but email as well, virtually any time of the day. So, enact your inner grace and just sort of feel free just to message anything that comes to your head, because I promise it will help. So any questions, please get in touch. But if I don't see you, have a lovely weekend. No, why would I see you? How silly. I won't see you. So have another lovely weekend. Bye-bye.